Hello, welcome back to my channel, Bio with Cheryl Shansi. In today's lesson, I want us to learn about osmosis. We are still covering topic number 4, chapter number 4 of the AS Cambridge syllabus, which is about cell membrane and transport. So, what is osmosis? With osmosis, whenever someone talks about osmosis, all you have to think about are water molecules. We can have our cell surface membrane. We know it is partially permeable, and sometimes cells need water. So we have our cell surface membrane here, which is partially permeable. So remember, a cell has cytoplasm. So if these are water molecules inside the cytoplasm of a cell, we can also have tissue fluid surrounding the cell with the water molecules in the tissue fluid. So we are going to have water molecules tending to diffuse from their region of higher water potential. So we are having two regions. I have a region inside the cell. Let me label that region B. And I have a region outside the cell. Let me label it A. Then I have my water molecule. So whenever we are looking at osmosis, it's all about movement of water molecules. In this case, we are going to have water molecules always tend to move from where they are many to where they are less. And that takes us to the term water potential. So when you look at these two regions, region A has more water. So we can say that region A has a higher water potential. So water potential can be written with this symbol, has a higher water potential. And then region B, which is inside the cell, has a lower water potential because it has less water. So with osmosis, we are going to have net movement of water molecules from a region of higher water potential to a region of lower water potential. In this case, from region A to region B. But for osmosis to occur, we always need a partially permeable membrane. The water molecules have to be crossing a partially permeable membrane. So osmosis is like a special kind of diffusion where we have net movement of water molecules from a region of higher water potential to a region of lower water potential. Please always use the term water potential because this is a common mistake by students to use the word concentration. So when you use the word concentration, sometimes it doesn't bring out it doesn't bring out osmosis the best way. So it's always better use the term water potential, that water molecules are going to move from their region of higher water potential to their region of lower water potential. Remember, it's net movement. Net movement means that we can have water molecules moving both sides from B to A and from A to B, but generally, more water molecules overall, more water molecules are going to move from A to B than from B to A. So we're going to have net movement of water molecules from A to B. So that is what osmosis is about. And so when you look at the definition of osmosis, we can define it as movement or the net diffusion of water molecules. It's a special kind of diffusion. Why a special kind of diffusion? Because we are still looking at water molecules moving from where they are more to where they are less. Just like in diffusion, where molecules move from the region of higher concentration to the region of lower concentration. So osmosis is the net diffusion, or even if you say the net movement of water molecules from a region of higher water potential to a region of lower water potential. And always osmosis occurs via a partially permeable cell surface membrane. Now, you cannot talk about osmosis and you forget to mention about a solute and a solvent. Remember, water is a universal solvent. Like in this case, I have these water molecules, the blue ones, which is a solvent. We have a solvent, the water, and then we have these ions that have dissolved in water, which we can call as solutes. So different solutions can have different water potentials. Like in this case, I have solution A and I also have solution B. So a solution that has more water molecules, like you can see in this case, it has more water molecules. The blue ones are more in A than in B. So this solution, we can describe it as being dilute. Dilute because it has more water molecules. Or we can say it has less solute. Because the ions are less, as you can see, it has less solute. So it has a low solute concentration. When you look at region B is concentrated because it has less water molecules and it has more solutes. So when it comes to osmosis, we are going to have net movement of water molecules from a region of higher water potential. So dilute solutions usually have a higher water potential. So here we have a higher water potential than, so we are going to have a higher water potential than this side B because it has less water molecules. So if osmosis to occur, we have our partially permeable cell surface membrane is only going to allow water molecules to cross. Why? Because the water molecules are small in size. So they are going to be able to cross from A to B, net movement from A to B. That doesn't mean we cannot have movement from B to A. We can, but net movement is happening from A to B. So water molecules are moving from their region of higher water potential to their region of lower water potential. And we can also say that these water molecules are moving down a water potential gradient. Down a water potential gradient simply means they are moving from a region of higher water potential to a region of lower water potential. So let's look at another example where we have let's use this diagram. We are having two solutions, A and B, separated by a partially permeable membrane, as you can see. We have solution A and solution B. Whenever osmosis is to occur, we always have to identify which region has a higher water potential, which region has a lower water potential. Obviously, when you look at this diagram, you can see region A has a higher water potential because it has more water molecules. Region B has a lower water potential, less water molecules, and more solute. Solute molecules cannot cross. Why? Because they are large. But the water molecules are small enough, they can pass through the partially permeable membrane. Remember, a partially permeable membrane allows some molecules to pass through, but others not to pass through. So when osmosis occurs, we are going to have net movement of water molecules from A to B. That is why you see this arrow is bigger. It shows you that overall we have net movement of water molecules from A to B than from B to A. Now, when water molecules get to B, obviously the volume of the solution in B won't remain the same. That is why after osmosis has occurred, we see the volume this side has increased. Why has it increased? Because more water moved from A to B than from B to A. So we had net movement of water from A to B. That is why the solution volume this side increased, or the level increased. So osmosis occurs up to when we have an equilibrium. What does an equilibrium mean? It is going to occur up to when we have these water molecules evenly spread between the two regions, just like in diffusion. That is why osmosis is considered to be a special kind of diffusion. So when you look at when you look at those notes, that is what I've just been explaining. So I'm going to skip that. When you look at the term water potential. Someone would be wondering, why are we using the term water potential? So water potential is the measure of the tendency of movement of water molecules from one place to another. So if a region has more water, the water molecules are going to tend to move more compared to a region that has less water. So always we are going to have water molecules moving from a region of higher water potential to a region of lower water potential. And don't forget, water potential can be represented by this Greek letter psi. So whenever I find such a letter written, I'll know that is water potential. I'll know that that means water potential. We are going to look at examples of questions where that is used. Now, when you look at the water potential of a solution, it depends on two factors. Number one, it can depend on the amount of solute that has dissolved in water. If a solution has more solutes, like in this case, we have solution B, you can see it has solute molecules, and you also have the water molecules. A solution that has more solute will have a lower water potential. A solution that has less solute will have a higher water potential. So if a solution has a lot of water, it's going to be dilute with a higher water potential. A solution that has less water but more solute is going to be concentrated, so it has a lower water potential. So when you add more solute to a solution, you're going to be decreasing its water potential. What other factor affects water potential? The other factor that affects water potential is application of pressure. Let's consider still these two solutions. We have seen solution A has a higher water potential than solution B. Now, we are going to expect net movement of water from A to B because A has a higher water potential than B. How about if you apply pressure? Assuming you apply pressure on the solution in B. When you apply pressure on the solution in B, that pressure is going to be squeezing these water molecules, it's going to apply pressure on them, so they are going to move more. And that raises the water potential. The application of pressure to a solution raises its water potential. The water molecules are going to tend to move more. And in that case, we might have the reverse. We might have movement of water from B to A than from A to B. Why? Because the water potential this side has been raised by application of pressure. Like you can see, if you apply pressure on this solution B, if you apply pressure, its water potential is going to increase. So here we are looking at solution B after osmosis has taken place. An equilibrium has been reached, but then you come and apply pressure to solution B. So when you apply pressure to solution B, because of the pressure, now solution B will have a higher water potential than solution A. And then eventually you're going to have more net movement of water molecules from B to A than from A to B. So that is what you need to know, that pressure increases the water potential of a solution. Now, water potential can be measured, and there are units that are used, which are kilopascals. So from this, I want you to note that pure water or distilled water has the highest water potential because it has nothing dissolved in it. It has very many free water molecules that are not attracted to any solute. And the highest water potential is 0. This is the highest water potential. So any other solution is going to have a lower water potential, which is going to be in negatives. So the more negative the water potential, the lower the water potential. The more negative the water potential of a solution, the lower the water potential of that solution. An example, if a solution has a higher water potential of -10, and you compare it with a solution of -20, you can see negative 10 is closer to 0 than negative 20. So this solution of negative 10 has a higher water potential than a solution of negative 20. So if you have both solutions mixed in placed in a trough and separated by a partially permeable membrane, the one for negative 10 kPa, and you also compare it with the one for negative 20, if they are separated by a partially permeable membrane, we'll expect net movement of water from the one for negative 10 to the one of negative 20. Why? The one of negative 10 has a higher water potential than the one of negative 20. So just know that the more negative the water potential, the lower the water potential of the solution. So when you find such in an exam, don't get confused. Just know that the highest water potential is 0. So all water potentials of other solutions are in negatives. Okay, so let's answer some questions here. They are telling us in figure this is the figure. The system has come into an equilibrium. Osmosis has taken place and we have an equilibrium. So there is no net movement of water molecules. What can you say about the water potential of the two solutions? Yes, whenever the water has moved and we have an equilibrium, usually we are not going to have net movement. What does that mean? It means that the rate at which water molecules move from A to B will be the same as the rate at which water molecules move from B to A. So in that case, the water potential of the two solutions is equal. So the water potential of A is the same as the water potential of B. Then here they're asking, the solutions A and B are at equilibrium. Which solution, A or B, is more concentrated? More concentrated, we can look at the one that has more solute. However, because they have the same water potential, none is more concentrated than the other because they have the same water potential. Why does water not move from They're also asking, why does water not move from the more dilute solution to the more concentrated solution? It is simply because the solutions are at an equilibrium. So we are not going to have net movement of water molecules because their water potentials are now equal. Why do we need to learn about osmosis? Osmosis can be applied in real life. It can affect our cells. It can affect plant cells. And let's look at the first case where osmosis is going to be affecting animal cells. We can have animal cells, particularly red blood cells. They can easily be used to experiment osmosis. If you place an animal cell, in this case, a red blood cell, usually a red blood cell has a normal shape, a biconcave shape. Now, when you place it into a solution of pure water, whenever you hear the word pure water, the word dilute, just know that that solution has a higher water potential than the cytoplasm of the cell that has been inserted into that solution. So obviously, we are going to have water molecules move from this solution into the cell. The cell is going to expand because it simply has a cell surface membrane. As more water enters into it, water applies pressure to the cell starts bulging, growing bigger and bigger, and eventually the cell surface membrane will not be able to hold the pressure because of the water that has entered it, then it's going to burst. So that is what happens to an animal cell once you place it in a solution that has a higher water potential than it. When you look at situation labeled C, this red blood cell has been placed in a solution that is concentrated, a solution that has a lower water potential than it. So you're going to have water molecules moving from the cytoplasm of this cell. That's why you see these arrows are showing you. We are going to have net movement of water from the cytoplasm of the cell into the solution that has a lower water potential. And yes, the water molecules will cross the partially permeable cell membrane. Eventually, the cell is going to become shrunk because it has lost water. And then we can have this part B, where we have a red blood cell placed in a solution that has the same water potential as the cytoplasm of the red blood cell. In that case, we are not going to have net movement of water molecules. The water molecules that enter into the red blood cell will be the same as the ones that leave. So there is no net movement. As a result, this cell is going to remain normal. So usually your body works and ensures that the concentration of your blood or your tissue fluid is the same as that of the cells that it is surrounding. So your blood should have the same water potential. Your tissue fluid should have the same water potential as the cells it's surrounding. Otherwise, if it has a higher water potential, water will move from the tissue fluid into your cells, and then your cells will end up bursting. At the same time, if it has a lower water potential, your cells are going to lose water to that tissue fluid, and they are going to end up shrinking. And once they are shrunk, that means that their functioning is going to be affected. So that is why when you're thirsty, your body is trying to show you that, hey, the water potential of your blood is decreasing below that of the cells. And then if you have too much water, your kidneys excrete most of that water out of your body. Why? So that your tissue fluid and blood do not become more dilute compared to the cells they are surrounding, to avoid cells from either bursting or shrinking depending on the water potential. Okay, so let's try to answer these questions. They're asking us, if you look at these figures, which solution has the highest water potential? Highest water potential, look at the one that is losing water to the cell. In this case, it's going to be solution A, which is pure water. So it's the one with the highest water potential. In which solution is the water potential of the red blood cell the same as that of the solution? It will be in this solution B, because the red blood cell remained normal and it did not burst or shrink. How about plant cells? Are they also affected by osmosis? Yes, they're also affected by osmosis. And we are still going to look at the same situations. The only difference is that plant cells have a cell wall, a rigid cell wall surrounding them. So this cell wall prevents plant cells from bursting. You'll never find plant cells bursting like the animal cells. However, the same situation happens to the plant cells if you place them in a solution of pure water or distilled water, or a solution with a higher water potential. What is going to happen? The pure water is going to have a higher water potential than the plant cell. So water molecules are going to move from the pure water into the plant cell by osmosis via the partially permeable cell surface membrane. The plant cell is not going to remain the same. The vacuole is going to grow bigger. The cytoplasm is going to increase in volume. We are going to have the cell surface membrane pressed against the cell wall. Why? Because when the water enters into this plant cell, a pressure builds up, and that pressure is called Tagger pressure. The pressure exerted by water that has entered into the cell is called Tagger pressure. So Tagger pressure is going to build up inside this cell, and that Tagger pressure is going to start pushing cell contents towards the wall. So if we have the cell contents to push towards the wall because of the pressure that has built up, eventually you're going to have the cell surface membrane touching the cell wall. Like in this case, this is the cell surface membrane, but it is touching the cell wall. And in such a situation where we have a high Tagger pressure, where the vacuoles are large, the cell has a high volume of cytoplasm, the cell contents are pushed towards the cell wall, the cell membrane is touching the cell wall, such a cell is described as turgid. Remember, these words are only being used in plants. So I shouldn't find using the word turgid in animal cells, no. So the cell is turgid, the cell has a high Tagger pressure. However, when this same cell is placed in a solution with a lower water potential, maybe a sucrose solution or a salt solution, the salt solution is going to have a lower water potential than the plant cell. So that is how water is going to be lost. We are going to have net movement of water from the plant cell into the concentrated solution. You can see how the vacuole has changed in size. The vacuole is now smaller. This is the cell surface membrane, but it has pulled away from the cell wall. This side it was touching the cell wall, but here it has pulled away from the cell wall. And now we are going to have space left here. We are going to have space left here. And usually the solution outside can enter and occupy that space. So such a cell that has a very small vessel that has lost water, the target pressure is decreasing or it has decreased. The cell membrane has pulled away from the cell wall. Such a cell is said to be plasmolysed. And plasmolysis occurs in plant cells. You shouldn't use the word plasmolysis in animal cells. We can also have these plant cells placed in a solution that has the same water potential as the cell. In that case, we shall have no net movement, which means the amount of water that leaves the cell to the outside solution will also be the same as the amount of water that leaves the outside solution into the cell. Why? Because the water potential outside this cell is the same as the water potential inside the cell. So there is no net movement of water molecules. So that is how osmosis affects plant cells. We have seen plant cells are surrounded by a very rigid, strong cell wall. So if water enters into the plant cell, usually plant cells never burst. Why? Because of that cell wall. So the cell wall is going to be resistant to expansion, meaning that even if the cell surface membrane tries to extend, it will finally touch the cell wall and the cell wall will prevent it from expanding any further. That is why there is no bursting. At the same time, we have a new term here, which is called protoplast. So when someone mentions about protoplast, it simply refers to the living part of the cell. Protoplast is usually used when we are looking at plant cells. So when someone talks about protoplast, we are looking at the cytoplasm of the plant cell and the cell surface membrane covering the cytoplasm. So it includes the cytoplasm, all the organelles, and the cell surface membrane. So when a plant cell has water entering into it, eventually the plant cell becomes turgid. Remember, we said, what does the word turgid mean? It means that water has entered into that plant cell. So the plant cell has a high turgor pressure. Spelling of the word turgor may be, this is the spelling of the word turgor. It has a high turgor pressure because water has entered into it. It is fully inflated with water. It has a high turgor pressure. It has a large vacuole. It has its protoplast, the cytoplasm and the cell organelles all pushed towards the cell wall. It has its cell surface membrane touching the cell wall. That is a turgid cell. That is a turgid cell. So let's look at this illustration. It shows us how plasmolysis occurs. So in this case, we are looking at a plant cell that has been placed into a concentrated solution or a solution with a low water potential. This is the cytoplasm. This is the cell surface membrane. Here it has been dotted so that you can easily distinguish it from the cell wall. This is the vacuole. So once this cell is placed into a solution that is having a low water potential, it's going to start losing water by osmosis to the outside solution. So since it's losing water, we are going to have the contents, the cytoplasm becoming smaller. Here they are showing you the cell surface membrane, hope you can see that it's starting to pull away from the cell wall. So that means that the process of plasmolysis is starting to occur. The protoplast is starting to shrink. That is, the cytoplasm is reducing, the vacuole is becoming smaller. You can see it's not the same size as it was earlier. And here the cell is fully plasmolized. Fully plasmolized means that we can see the cell surface membrane has totally detached from the cell wall. The vacuole is so small. This cell is going to have a very slow Turger pressure, a very low Turger pressure. And we have space within this cell. So eventually, remember I told you that eventually, if this was our external solution, the external solution is going to start entering into this cell. Why? Because remember the cell wall is fully permeable. So the solution will cross the cell wall and come to occupy this space, to come and occupy that space. So here the cell is fully plasmolized, and plasmolysis makes plant cells wilt. Why? Because some plants derive their support from turgid cells. So if the turgid cells have become plasmolized, then the plant will no longer have support. So its cells are going to be plasmolized, and then since it has no support, that is how wilting is going to occur within that plant. So I have been able to explain all this. I've been able to explain all this. Yes, all this. So maybe I've not talked about this term, incipient plasmolysis. Incipient plasmolysis simply talks about that time when plasmolysis has started to occur. Like in this case, we are having the protoplasts, or we are having the cell membrane starting to pull away from the cell wall. So that is what the word incipient plasmolysis means. Okay, so certain key terms to remember. We have a protoplast. Protoplast means the living content inside a plant cell. That is the cell surface membrane and all that is inside the cell surface membrane. Plasmolysis is where we have loss of water from a plant cell or a prokaryotic cell like bacteria, where the protoplast has shrunk away from the cell wall. Incipient plasmolysis means the point at which plasmolysis is about to occur, where we are having the cell surface membrane starting to pull away from the cell wall, and the Turger pressure has really decreased in that the cell membrane is no longer touching the cell wall. Let's look at this question. We are having two neighboring plant cells shown. We have plant cell A and plant cell B. And these two cells have different water potentials. So the question is, which plant cell has the highest water potential? If those cells are neighboring each other, which of the two cells will lose water to the other? We are going to have net movement of water from which cell to which cell. Okay, if you have been following, you know that obviously water potential can be measured in kilopascals. And we saw that the more negative the water potential, the lower the water potential. So that means cell A has a higher water potential. So if osmosis is to occur between the two cells, we are going to have net movement of water molecules from cell A to cell B, then from B to A. So you're going to have net movement of water molecules from A to B. So basically that is it about osmosis. We shall look at some sample questions, but when it comes to experiments that you do in your lab, you can be expected to investigate osmosis in plant cells and then observe them under a microscope and observe the different stages, some cells that are turgid, or those that have gone through plasmolysis. Then you might use potato tissue to show osmosis, or you could be expected to measure the length of the tissues or the mass of the tissues. So all those are different experiments that you'll be carrying out in your during your journey in your labs. But just know that an example, if I have potato tissue, if I see that tissue has grown longer or it has gained mass, it's simply an indicator that the plant cells were able to have water enter into them by osmosis, meaning they were in dilute solutions. If I see the plant tissue has become smaller or it has lost mass, it's simply an indicator that that plant tissue was in a solution of lower water potential, so it lost water to the solution. So that is it about osmosis. Thank you for your time. If this video helped you, please give it a like, subscribe, share with your friends. See you on the next one. Bye.